Hi, my name is Zach Haig, and thank you for taking the time to watch my video. 200 years ago, bacterial infections caused the majority of deaths worldwide. The rise of antibiotic resistance could plunge us back into a pre-antibiotic era if it goes unsolved in the near future. So in this presentation, I will be discussing a potential plan to combat antibiotic resistance through the use of quorum-sensing activated bacteriophage therapy to fight methicillin-resistant Staphylococcus aureus infections, also known as staph infections. Now, before I go into detail on the overall process, I will discuss and explain some of the individual components involved. So antibiotic resistance ultimately arises from mutations in the bacteria that create new genes that forms an intolerance to antibiotic drugs. It is projected that this could cause 10 million deaths per year by 2050 if it goes untreated. And in addition, there are no new sources of antibiotics out there right now. This shows that we do need some new alternative treatments for bacterial infections. The figure on the right here shows some common causes of antibacterial resistance, the main ones being misuse, overuse, and incomplete treatments by patients. Methicillin-resistant Staphylococcus aureus, also known as MRSA, is a specific strain of antibiotic-resistant bacteria. It is completely intolerant of methicillin, and it is also partially intolerant to other drugs as well. So far, it has caused 20,000 healthcare-associated infections here in the US, and there is no sign of that decreasing in the near future. The figure on the right here just shows some MRSA cells under a microscope infecting epidermal skin cells, since it is a surface infection. Bacteriophage, also known as phage, are bacterial viruses that infect bacteria in one of two ways. The first is a lysogenic pathway, shown on the right here, where the phage inserts its DNA into the bacteria and manipulates the cell from the inside. The second is a lytic pathway, where the phage breaks the cell membrane and kills the cell on site. It has been shown that this can be used as a therapy to fight antibiotic resistance and also to fight off bacterial infections in general as well. It can do this by either turning off the resistance genes to allow antibiotics to work effectively, or it can replace the antibiotics and directly attack the infection. Quorum sensing is the communication mechanism of bacteria. And it is specific to each strain of bacteria, although a study has shown that it can be synthetically inserted into other bacterial strains, such as E. coli, so that the E. coli can communicate with MRSA through quorum sensing. The process of quorum sensing is summarized in the figure on the bottom there. The first step is synthesis of the signaling molecule by a cell. Then the second step is the recognition of that signaling molecule by the surrounding cells. And the last step is the response of those surrounding cells through gene regulation. This can be the virulence factors, which are the infectious and dangerous parts of the cell, or it can be smaller things like motility and bioluminescence. Now, as I said, these are all individual components of the overall project idea, which is summarized here on this slide for you. So the first step is to apply the phage therapy as an ointment on the infection site. Then the MRSA cells will release their signaling molecules, which will be recognized by the ointment itself. This will trigger the quorum sensing inside, which will release the phage from the ointment. The phage will then attack the virulent MRSA cells, insert their DNA, and deactivate the virulence factor genes within the cell, making the cell safe once again and ultimately killing off the infection itself. This also has an advantage over antibiotics because it does not affect the natural bacteria of the body, which causes a lot of fatigue and susceptibility to future diseases in antibiotic use. This process is currently going through a feasibility study, and if that is successful, it will move on to some research trials, clinical trials, and then into healthcare to fight antibiotic resistance. I hope you found this presentation both exciting and informative, and thank you again for watching.